This is chapter 24 that deals with metabolism and nutrition. Metabolism is simply the complete sum of all the chemical reactions that are occurring in the body. The rate of metabolism will depend on several different things. It's going to depend on age. Typically, as you get older, metabolism slows down. Depends on gender. On average, males usually have a faster metabolism than females do. What's your activity level? What's your consumption of food like? What's your body mass? Genetics is also going to play a role in it. When we talk about the metabolism, as I just said, it's a sum. So it's going to be a combination of both catabolic reactions and anabolic reactions. Catabolic reactions are those where you are taking large molecules, breaking them down into smaller molecules, and that's going to release energy. Anabolic is the reverse. It's where you're taking smaller molecules and you're going to connect them together to form larger molecules, and that is going to require the input of energy. One thing to keep in mind when we talk about metabolism and these various types of reactions is whenever you hear ATP, adenosine triphosphate, that is basically energy. Think of energy at the cellular level whenever you see ATP. And this is just a schematic drawing to show, as I just said, with anabolic reactions, taking those smaller molecules, you're going to form bonds, connecting these together. And so that's going to take energy to connect them together. But the reverse of that, which is a catabolic reaction, there's energy stored in those bonds. That's why it required energy here, is you had to put energy in there to connect them together. Well, now when you break it apart, and you break that bond between these components, that releases the energy. And so usually these two types of reactions are tied together, because where does this energy come from that you have to put in to make the bonds? Is coming from a catabolic reaction. So the energy released here is used up here. And this is just another way of showing for ATP, adenosine triphosphate. You have this adenine and the ribose, this complex right here, adenosine. It says triphosphate. It means you have 3 phosphate groups. These bonds here, these phosphate bonds between them, are very high energy. So when you break one of those apart, it's going to release a lot of energy. And so you have ATP. You take off this outermost phosphate, break the bond right here, release the phosphate, and now you have ADP, adenosine diphosphate. When you have extra energy, extra phosphate here, you can put it back on. This reaction is reversible. So once again, ATP, energy for the cell. And so in this chapter, what we're going to very briefly look at are some of the various different types of reactions. You eat your meals, snacks, intake that food. How do you break it down? Now we're going to focus primarily on the carbohydrates and how your complex carbohydrates are going to be broken down into simple sugars such as glucose, and then that's broken down to produce ATP so that the cell has energy. We'll also briefly look at the proteins and also the lipids and how they're broken down. But notice one thing from here. Proteins can be broken down to amino acids, which can then come in and feed into part of the reactions from carbohydrates. Same thing for triglycerides, those lipids. Some components can feed into, look at this, a common factor here, and ultimately help produce ATP. So if we look at the carbohydrate metabolism first, we talk about polysaccharides. Those are complex sugars. Those are things like starch and glycogen and cellulose. The body can't use those. They're way too big. Not at the cellular level. It can't take it across that cell membrane. So it's going to have to be broken down into monosaccharides or simple sugars, things like glucose, fructose. And I'll tell you right now, any cell, whether you're talking about a human cell, a bacterial cell, a fungal cell, all cells are able to use glucose. Now, some of your other types of carbohydrates, sometimes yes, sometimes no, but they can all use glucose. And that's going to be the preferred choice. if a cell is given a variety of different things, they're always going to go for the glucose first. It's the easiest thing for it to break down. Cellular respiration has nothing to do with your breathing. What cellular respiration is, the breakdown of sugar at the cellular level. It is taking glucose, a simple sugar. It requires oxygen for the entire process to occur. There are going to be multiple steps, different phases of it, but for the entire thing to occur, oxygen will be required. It will break it down, the glucose, which is a 6-carbon sugar. It will break it down to carbon dioxide, which is a 1-carbon compound, and water, and ATP. And a lot of people forget about this component of the reaction. This is the whole reason why you need all those nutrients. You need the ATP. You need that energy in order to run all of the various metabolic processes within the cell. So cellular respiration, as I just said, it's divided into different phases or different stages. It's going to always start with glycolysis, and then you're going to have the Krebs cycle, and then oxidative phosphorylation. So there's a lot on this slide. You start with glycolysis. Glycolysis is taking that glucose, which is a 6-carbon compound, and you are going to break it down to pyruvate. You're going to actually break it down to 2 pyruvate molecules because pyruvate is a 3-carbon compound. In chemistry, one of the most fundamental basic laws is you can't just suddenly create things. So if you start with 6 carbons, you have to account for all of those 6 carbons. So we tend to, with metabolic reactions, compare things like per glucose molecule. So yes, we're going to take glucose. We will break it down to 2 pyruvates. Initially, you're going to require some ATP, and then you generate some, but your net gain from glycolysis is 2 ATPs. You will generate also 2 NADHs. This is a form of an electron carrier, and we're going to see that again down here in the last step. So that's why we're counting. For this part of the process, oxygen is not required. So glycolysis will occur whether oxygen is present or not. Now the next, and just so you know, I have a diagram to show you this. This is glycolysis. That's actually about 10 different steps. So it doesn't just cut it in the middle. We'll go over it in a moment. So this is just kind of a summary. The next step, and in our cells, glycolysis occurs out in the cytoplasm. The pyruvate is going to have to be moved across the mitochondrial membrane and into the interior portion of the mitochondria, excuse me, where the rest of the process is going to take place. The Krebs cycle, which is also known as the citric acid cycle, which is also known as tricarboxylic acid cycle. Microbiology, we tend to call it the Krebs cycle. Biology calls it the citric acid cycle. Chemistry calls it the tricarboxylic acid. It's all the same thing. And here you're going to break down that pyruvate. Once again, a net gain of 2 ATPs. You'll have 6 NADHs, 2 FADH2s. These are also an electron carrier, which we're going to see in the next step. And this also does not require oxygen. However, it will not occur if oxygen is not present. It doesn't require it, but the next step, oxidative phosphorylation, does require oxygen, and it's one of those things, if you went through the Krebs cycle and there's no oxygen, then you're stuck with these, and you're at a dead end piling up all the stuff you can't do anything with. So why even bother? So we'll see what happens in a moment. If oxygen is present, fine, everything continues on from glycolysis on down through the Krebs cycle, and then on to oxidative phosphorylation. This is where most of the ATP is going to be generated. And this includes, it's often divided into 2 parts, the electron transport chain, and that is where oxygen is required. It will not occur without oxygen. And then the final step is chemiosmosis. As we'll see, is the process of helping make the ATP. And this is where the NADHs and FADH2s, those electron carriers, come into play. For every NADH that you generated in these steps up here, for each one, you will generate 3 ATPs. And for the FADH2s that were created here, for each one, you will generate 2 ATPs. For glycolysis, the first step, we're going to take glucose, which, as I said, is a 6-carbon compound, and we are going to break it down into 2 pyruvate molecules. And that is, if you break it down, it's about 10 different steps. So we're going to start up here with the glucose. Now, initially, you have to put some energy into it. So that's why it's called energy consuming phases. You're going to actually use up 2 ATPs initially. So you haven't gained anything. You're at a loss right now in terms of energy. You're going to put that in, add phosphates on the end of these compounds, and then finally break it down into 2 3-carbon compounds, shuffle around the phosphate groups, and then eventually end up with 2 pyruvate. So this is one way, and this is in your textbook, the way they have it drawn out. And it's not until down this last part where you actually are releasing some energy in the form of ATP. This is another way of looking at it. Some people like to see the compounds drawn out like this. So you're going to start off with glucose. You have to add ATP. You take off one of those phosphate groups and you stick it here on the end. And then that's called glucose 6-phosphate. The numbering in chemistry, the 6 refers to, it's on the number 6 carbon. We number the carbons down, and so it just means you put a phosphate on the number 6 carbon of glucose. And then it's converted to fructose 6-phosphate. Fructose and glucose are both 6-carbon compounds. The difference is right up here where you have this oxygen and this OH. You just swap those places. Then you take another ATP, take a phosphate group off of it, and stick it on the other end. So now you have fructose 1,6-bisphosphate. Now you're going to separate those into, split it right here into 2 3-carbon compounds. And these can alternate between the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now you're going to have NAD, which is this electron carrier. It's going to come take off a pair of electrons, and you have NADH. And this is what we will see later with the electron transport chain. In the process, you've added another phosphate group, so you have 1,3-bisphosphoglycerate. Glycerate, for those of you who have not had chemistry, is a 3-carbon compound. So anytime you see glyceraldehyde, glycerate, glyceric acid, glycerol, all of those means it has 3 carbons to it. And the 1 and 3 is telling you where the phosphates are. You remove one of those phosphates, stick it on ADP. So you just stuck another phosphate on it, making ATP. You're left with 3-phosphoglycerate. Shift that from the number 3 carbon to the number 2 carbon to 2-phosphoglycerate. Remove the water. End up with phosphoenol pyruvate. And then take off the last phosphate group, stick it on ADP, forming ATP, and you end up with pyruvate. Excuse me. This is a basic, a fundamental step that all cells are capable of carrying out. And so as you go into more advanced classes, you have to know all of these compounds. And in an upper level course, you have to know all the enzymes, which are the enzymes that are catalyzing these reactions. That's what's written in these blue colors here. You don't need to know that. Excuse me. And just so you know, I realize some of you have not had chemistry, and if you're wondering, why couldn't you take this 6-carbon compound back here? And man, I could just cut it into, straight to 2 3-carbon compounds. Why do you have to do all this rearranging? well, chemistry, you have to realize this is just drawn on paper, but these are all actually three-dimensional compounds. And so it's not always as easy as just cutting it in half and breaking the bond right here. And the analogy that I usually use is it would be similar to going on a field trip and a kindergarten teacher going with a bunch of kindergartners on a bus. Okay, everybody get on the bus, 2 kids per seat. Well, then you look at them and you go, oh, wait a minute. We can't have little Joey and little Mark sitting next to each other because they're going to end up fighting. So you got to move Mark over here. Oops, but no, now that puts him behind Susie and he's pulling her hair. So we need to move Susie over here. And oops, Mary threw stuff out the window on the last field trip, so she can't be by the window. We got to move her over to an aisle seat and, all that shuffling around. It's kind of the same type of thing here, that you can't just necessarily just cut it in half, because sometimes some of these players right next to each other are like 2 little 5-year-olds next to each other on the school bus that don't get along. That you have to be very careful how you cut things, realizing that these are three-dimensional compounds. So you just go through the steps: glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, to glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenolpyruvate, and then pyruvate. This is glycolysis. This occurs in the cytoplasm. Now, the pyruvate is a 3-carbon compound. We're going to move to the Krebs cycle, but we're starting with acetyl-CoA, which is a 2-carbon compound. So what happens to the pyruvate? Well, number one, the pyruvate was in the cytoplasm. The Krebs cycle occurs in the mitochondria. So we have to move that pyruvate across the mitochondrial membrane inside the inner portion of the mitochondria. That's going to cost you some ATPs, 2 ATPs. And then that 3-carbon compound of pyruvate, you need to take or remove one of the carbons off. It's going to leave a CO2. And you have coenzyme A come in and combine to those other remaining 2 compounds, forming acetyl-CoA. So acetyl-CoA will now feed into the Krebs cycle, but pyruvate does not directly. You have to take one of those carbons off first. So now you have acetyl-CoA coming in to the Krebs cycle. Acetyl-CoA will bind with oxaloacetate, which is a 4-carbon compound. So you take those 4 carbons, combine the 2 carbons from acetyl-CoA, and you just made another 6-carbon compound called citrate. Shuffle things around a bit, still 6-carbon compounds, isocitrate. Here's our good friend NAD coming in to pick up a pair of electrons, leaving as NADH. Take off a carbon, leaves a CO2. You're left now with 5 carbons, and it's alpha-ketoglutarate. Here comes another NAD, takes off a pair of electrons. Here goes another carbon, leaving a CO2. Now you're down to 4 carbons, and it's succinyl-CoA. Do some shuffling around. You've got a phosphate group here. Look this, you just generated an ATP. Succinyl-CoA becomes succinate. Here comes the other type of electron carrier. FAD comes in, picks up a pair of electrons, leaves us FADH2. And remember, the FADH2s, the NADHs, we're going to account for those later. We're going to see them again. You're left anyway with fumarate. The water come in, shift around, and you now have malate. Here comes another NAD. Excuse me, picks up a pair of electrons, leaves this NADH, and you now are back to oxaloacetate. Now you had 2 pyruvates that you generated from glycolysis. We're going to base everything per glucose molecule. So if you had 2 pyruvates, that means you go around this 2 times because you had 2 pyruvates feeding into this. Notice that they have all these other components here showing you the relationship of how, not only for carbohydrates, but fatty acids and steroids, those are forming lipids. Amino acids here for forming proteins, nucleotides for forming nucleic acids. Aspartate is an amino acid. Forming proteins can also be used for making nucleotides for your nucleic acids. In microbiology, remember pyruvate was right here. In microbiology, we have a phrase that we use that all roads lead to pyruvate because pyruvate is such an instrumental component for metabolism. So many reactions either start or leave from there. So you notice that oxygen was not required here. However, if oxygen is not present, it won't even waste its time because it cannot do anything with these NADHs or FADH2s if there's no oxygen. So why make all of this just to have it like stockpiled? No, that makes no sense. The cell's not going to do that. So although you don't need oxygen directly for any of these steps, the next step will require oxygen. So if oxygen is not present, it's not going to go through this. There's an alternate route that it will take. Excuse me. And once again, this is just another diagram showing. Here is the pyruvate, and this is what I was talking about, how you're going to remove one of those carbons that leaves as carbon dioxide. You are left with a coenzyme A coming in, combining with those two carbons, making acetyl-CoA. And now this is what feeds into the Krebs cycle, which is once again shown here. For those of you who like to see the compounds drawn out, you can see them obviously here. Oxidative phosphorylation. We are still in the mitochondria. This is where you are going to generate most of your ATP. And this is the steps. It's divided in two parts. You have the electron transport chain and then what's often referred to as chemiosmosis. The electron transport chain is this portion right here in red. And then chemiosmosis involves the ATP synthase, which is this blue structure right here. If you remember, the mitochondria has two separate layers of membranes. This is occurring in the innermost membrane. So what happens here on the oxidative phosphorylation, here's your NADH. It is carrying, remember, those pairs of electrons. It's going to drop it off on this protein. And we know the names of all these proteins. You don't have to worry about it, but we do know the names, exactly how it works. So it's going to drop off the pair of electrons here. It leaves this NAD. It can go back and be reused again. Years ago, I taught from a book, and they described NADH and FADH2s as taxicabs, which initially I thought was kind of crazy, but then the more I thought about it, it's like, well, it kind of makes sense if you want to think of it that way. They're both electron carriers. They both will pick up electrons, and they always pick up a pair. So yeah, it is kind of like, you know, the taxi cab. It picked up its two passengers. Where does it take it? Well, it's taking them, let's say, to the airport. Well, if you're on, you know, one airline, you drop off your passengers here. And what do? You go back out on the road to pick up more passengers. And that's what the NADH is going to do. Drop off its pair of electrons, and then it's going to leave and go back, pick up another pair of electrons. What happens to these electrons is that they get shuffled. That's why we call it a transport chain. These pair of electrons will get passed over to this Q, which then is going to pass the electrons over here, which then passes them to here, which then passes them to here. So it's passing it down this chain. Every time it's passing these certain landmark proteins, these integral proteins that extend the entire width of this membrane, inner membrane, it's going to move hydrogen ions from the inner matrix into this space here, but the other membranes out here. So it's going to move it. And then here you pass the electrons here, and it's going to move some more hydrogen, and then some more hydrogen. So what you are doing here is you're making a concentration gradient. You're taking the hydrogen ions here and moving them over here, so now the concentration is getting very high over here. Now what happened to those pair of electrons down here at the very end? That pair of electrons, guess who the final electron acceptor is? Oxygen. That is why oxygen is required for this step. It's going to be the last thing picking up those electrons. And then it's going to combine with two hydrogens and it's going to form water. And if you remember from the equation for cellular respiration, your final products, you started with glucose, plus you needed oxygen. This is where you need the oxygen. And what do you end up with? Water. How? Because right here the oxygen combines with the hydrogens, forming water. Plus CO2. Where did the CO2 come from? Remember, as you're breaking down the glucose and the pyruvate, every time you took off a carbon, how did it leave? As CO2. So you're producing this water here. Okay, well, you've been moving these hydrogen ions over here every time you move the electrons. And by the way, here's FADH2. It's dropping off its electrons at a different, if you're talking about airlines, it's a different airlines. It's a little bit further down. And so, same thing. Transfer the electrons here. They get passed along. It doesn't go as many spaces. And that's going to play a little bit role in a minute. So what happens to all of these hydrogen ions? You're building up all these hydrogen ions over here. Well, this ATP synthase, what it does is it allows the hydrogen ions. Remember, things want to go from high concentration to low concentration. Well, if you've been shifting all these hydrogen ions out, and some of them are combining with the oxygen here to form water, you have now low hydrogen concentration here, high on this side. So they're going to naturally just flow through this enzyme. This is like a turbine. And that's what it's going to generate. You have ADP and you have phosphate. Stick the phosphate on the end, generating ATP. So this electron transport chain, you're moving the electrons along. Ultimately, oxygen is the final electron acceptor, and then it combines with hydrogen to form water. And over here is where you are generating all the ATP. For every NADH, you will generate 3 ATPs. For every FADH2, because it comes in a little bit further down, you will only generate 2. So let's go back and look at the entire cellular respiration. And what did we generate? If we're talking about the whole purpose of this is to generate ATP for energy from glycolysis itself. Yes, you generated 4, but at the very beginning you required 2, so your net gain is only 2 ATPs. You also generated 2 NADHs. Well, when you had to convert pyruvate to acetyl-CoA, you generated 2 NADHs. So now you're up to 4. In the Kreb cycle, remember, you generated 2 ATPs. You generated 6 NADHs and 2 FADH2s. All of these were transferred to the electron transport chain to drop off those electrons. So you had a total of 10 NADHs and a total of 2 FADH2s. So if the NADH, each one generates 3 ATPs, that's a total of 30 ATPs, and the FADH2s generate 2 ATPs, that's a total of 4. So that means the electron transport chain by itself, just that component, you've generated 34 ATPs. Now, you have 2 ATPs from the Kreb cycle, so that brings you to 36. And then you have 2 ATPs from glycolysis, which brings you to 38 ATPs. But remember, I said it was going to cost you 2 ATPs to move across the mitochondrial membrane. So for a eukaryotic cell, for animal cells, such as our cells, per glucose molecule, you will get 36 ATPs per glucose. Just so you know, bacteria, which are prokaryotes, which don't have a mitochondria, and they carry all of the same processes out, but in their cell membrane. But because they don't have a mitochondria, they have to transport the pyruvate across. They don't lose 2 ATPs here. So they actually generate 36 ATPs per glucose. But for your cells, cellular respiration, 36 ATPs per glucose. So what happens if oxygen is not present? Well, then the cells can go through anaerobic respiration. And you still go through glycolysis producing the pyruvate, but when oxygen is not present, like I've said a few times, it's not going to lead to acetyl-CoA and feed into the Krebs cycle because it can't do anything with those NADHs and FADH2s. It's a waste. And so it doesn't even flow that way. It's like a fork in the road. If oxygen is not present, it will carry out anaerobic respiration, meaning it will take that pyruvate, and in our cells, it will produce lactic acid. Cells either will go through acid fermentation or alcohol fermentation. And certainly for humans, what we have is lactic acid fermentation. A lot of microorganisms carry out alcohol fermentation, which we have learned to capitalize upon that. And it's probably a very good thing that humans do not carry out in our own cells alcohol fermentation. What's the advantage? Well, in the absence of oxygen, you can still generate 2 ATPs, which is not much compared to 36, but it's better than nothing, so it will still keep you going. So which is more efficient? Cellular respiration by far, because you're getting 36 ATPs. But the advantage of anaerobic respiration is that it can be very effective for a very short period of time until you can get more oxygen, like when you're exercising. It allows you to still produce some ATP until you can get your respiration rate up, your heart rate up, to get more oxygen to the muscles so that they can then switch back and carry out the cellular respiration. Gluconeogenesis is the process of producing glucose, and this usually occurs in the liver. And it will occur, why do you need, well, if you need glucose for things, glucose levels are really low, and you're trying to bring those levels up. So if you're in a situation, why would glucose levels be really low? Well, if you're fasting, this will occur. If you're on a very low carbohydrate diet, this will occur. And certainly if you're under a starvation situation, then this will occur. Given a choice at the cellular level, it will go for carbohydrate metabolism first. It will go through glycolysis first. Lipid metabolism. This is associated with carbohydrate metabolism because a lot of the precursors are often going to be carbohydrates. Triglycerides are going to be broken down into fatty acids. You get more energy per unit mass with lipid breakdown than you do with carbohydrates and proteins. And protein metabolism, proteins, remember, are involved with every single cellular process that's occurring. And so there's lots of different types of proteins, there are lots of different types of functions. When you have extra proteins, though, they're not stored. Instead, they're usually converted to glucose or triglycerides. There are 20 amino acids. Remember, amino acids are the building blocks of proteins. Not all of them are available. Do we synthesize? So some of them we must get through our diet. So in terms of at the cellular level for energy sources, the primary source at the cellular level is going to want to use carbohydrate. And given a choice between a selection of carbohydrates, the cell's going to use glucose first. If carbohydrates are not present, then it will go for lipids and start breaking the lipids down. If carbohydrates and lipids are not present, then it will start to break down protein for energy. But at that point, you are in trouble. If there's no carbohydrate and there's no lipids around to use, and the only source for energy you have is protein, you are in trouble. This is one thing that happens under severe starvation conditions. You may or may not survive. This chapter also looks at nutrition and diet. So very briefly, when you're talking about nutrition, we often refer to calories, and this is with a capital C. This is the amount of heat it takes to raise 1 kilogram of water by 1 degree Celsius. You need, on average, 1,500 to 2,000 calories per day to sustain all of the processes that are occurring. Now, how much you take is going to depend on your body mass, your age, height, gender, level of activity, the amount of daily exercise. In general rule of thumb is for every extra 3,500 calories, that's going to equal a gain of 1 pound. And unfortunately, a lot of us don't realize how many calories we are taking in, and found it interesting and thought about, yes, it's true. Also, your book mentioned that a lot of people underestimate the amount or the ratio of calories versus exercise. They think they're taking in fewer calories and they're exercising more than they, and therefore burning more calories than what they're doing. And this is from a nutritional standpoint. The guidelines of what your diet should be like, they keep changing this. I remember when I was first in school way back in the dark ages, we had the food pyramid, and now they have changed it to the My Plate version, basically saying half of your diet really should be fruits and vegetables. The other half, the majority, or it's not quite 50-50, it should be more grains and limit the protein. And then also you have your dairy and fluid intake. Vitamins are organic compounds. They function as cofactors, so that means that they assist enzymes with their metabolic processes. Enzymes, remember, are catalysts that will speed up the rate of reactions. Well, sometimes they need helpers. Cofactors would be, a cofactor is a vitamin, a coenzyme is a mineral. So vitamin is organic, meaning it has carbon and hydrogen in it. Vitamins can be classified as either fat soluble or water soluble. The fat soluble ones are vitamin A, D, E, and K. Remember, E. coli in your large intestines helps to make vitamin K for you. Because these are fat soluble, if you take in too much, it does accumulate in the body, and I think everyone is familiar. If you don't have enough of these vitamins, there can be issues, but also if you have too much, it can also cause issues. So you have to be more careful with the fat soluble ones, that you're not taking in too much of it. The water soluble ones are all of the B vitamins like B12, folic acid, biotin, etc., and then vitamin C. Now, because these are water soluble, if you don't have enough, there can be abnormalities caused by not having enough of these vitamins, but there's not usually a problem with excess because that's usually going to be released in your urine. So some of these we synthesize, vitamin D, we synthesize some of that, but others have to be obtained from our diet. And then the minerals, as I mentioned, they act as coenzymes. Once again, it's one of the huge things that they will do, but they have a lot of different functions in the body. They're inorganic, so they don't have carbon in them. So we do not synthesize them. You must get them from your diet. You often don't need very much. We have the major ones, and we have trace minerals, which you only need minute amounts, but you have to get it from your diet. And this includes things such as potassium, sodium, calcium, phosphorus, magnesium, and chlorine. Chloride are the most common of those.